Today I'm going to talk about some of the recent work from my group and about how to represent whole genome polymorphism data at biobank scale and why we think this data structure could enable efficient analysis. So genetic polymorphism or genetic variant, as this audience should be familiar with, are extracted from this DNA sequence data, and then we get this polymorphism, and we can get this 0-1 representation of this polymorphism as what happens in each individual at a particular variant. And it could also be encoded in this integer state space as 0, 1, 2 to represent homozygotes, heterozygotes, and the other homozygotes. Haplotype phasing would be able to separate a diploid state which we don't know where these variants lie along a chromosome into these possible phases, and one most probable phase will be chosen. And this haplotype phasing at the biobank scale whole genome data is more or less a solved problem. So even though it's still computationally expensive, we now have access to this phased whole genome at the UK biobank scale thanks to methods like BEAGLE and ShapeIt. So this is readily accessible. So for today's talk, I'm mostly going to talk about how we want to deal with this phased data. So why do we think we need a different type of representation for this phased data? That's really because we're starting to have more than 1 million whole genomes with UK Biobank as the leading resource, but the other biobanks are also catching up. And conventionally, we use this type of tabular encoding, which is pretty much a matrix with this sample genome and this genetic variants at different positions. It's very intuitive, and you can also use highly optimized algebra library to do different types of computation, dot product, and different types of operation with it. The downside is with this large data set is not even possible to load this data into the RAM, and doing whole genome analysis can be very expensive. And another thing is this tabular format, you have to arrange it as vectors, for example, in RAM, and you can either arrange it by samples or you can arrange it by SNPs, and that allows you to extract the data very easily in one way, but not in the other way. And to be able to extract both sample data and variant data very easily, you sort of have to duplicate its representation in RAM in order to do that. So it's not like a symmetric way in doing these different tasks. So ideally, we're hoping to get a different representation that could improve computational scalability, and especially like sort of symmetric in terms of how we handle samples and how we handle variants, and also maximize the biological interpretability. And so we want this new data structure to sort of still reflect this data generating process biologically. And this is sort of related to this computer science concept about succinct data structure, that is talking about a data structure that's close to the information theoretic lower bound to represent the data, but could still compress the data, but still allow efficient data query. We're not claiming that we are making this succinct data structure, but we think we're working towards that direction. So intuitively, if species DNA sequence are related through a true evolutionary relationship and with only mutations causing their differences, a phylogenetic tree could be used to also represent these genetic differences that's caused by mutations along these branches. So a phylogenetic tree, on the one hand, is encoding this species relationship, and on the other hand, if everything is true, then it could also encode this DNA sequence alignment. With population level data that we deal with, there's recombination. So this relationship among different samples is no longer represented by a single tree, but at different regions in the genome, it could be different tree topology and a different relationship. But similarly, we have this concept called ancestral recombination graph, which is primarily used to encode this genealogical relationship. But as we can see here, these mutations happen along the branches, and from one tree to another tree. This is caused by recombination. So this graph could also be used to encode DNA sequences at these leaf nodes or the samples. So this is a great data structure that could be that's really great for encoding genealogical relationship, but it can also be used to represent DNA sequence data. However, it's not that easy to extract DNA sequence data at individual level or mutation level directly from this graph. And so this graph currently, with the development of tree sequence by Kelleher and colleagues, this tree sequence representation of this graph is actually a very compact way of presenting biobank-scale whole genome sequencing data. It's not currently possible to do it biobank-scale yet, but it is a really compact format. So the way this tree sequence works is that there are a few objects with mutations, and the sites are really just attributes to these mutations. And these mutations would have a node above this mutation, which would be, in this case, this node, and a node below this mutation in this mutation table. And through this mutation table, you can look up the nodes and look up the nodes that would lead to this edge that is between these two pairs of nodes. And with that, we can keep tracing the nodes below it and extract which samples have these mutations. So you can see the genetic data is fully encoded, but it's not exactly that easy to extract this information from tree sequence. And and then, of course, the downside I mentioned is like arg inference from DNA sequence data is actually a very difficult reverse problem. And we currently do not have any method that allow us to infer biobank-scale whole genome args, and extracting data is not exactly that easy. So with that, it motivates us to think about a slightly different way to represent this data, and which we call genotype representation graph. And so this is a small illustration of what kind of data structure we are thinking about. So in this case, we have 2 root nodes, that is 6 and 7, and we have 4 sample nodes from 0 to 3, and 5 to 6, 4 to 5 are the internal nodes. So we define root nodes such that they must contain at least one mutation. And we also define this graph as there exists at most one path from any given root node to any given sample node. And there exists one path from the mutation node containing a mutation to a sample node if and only if the genotype for that sample node contains that mutation. So I'll explain in an example what exactly this means, but this is roughly speaking the definition of this graph. So we want to encode the same data as a genotype matrix. And how do we get that information? So let's say this mutation 5 and 10 are the mutations we want to encode as an example. And we can see from this table that these 2 mutations, they share exactly the same information among these different haplotypes. Okay, so M5 and M10, they're here. They are on node 7. And how do we get the information about the samples from M5 to M10? So we traverse downward and to 0, downward to 4. 4 leads us to 2 and 3. And that gives us the information that 0, 2, 3 having the state of 1, that is the carrier of this mutation. And the default mode is they don't. So we know that sample 1 doesn't have this mutation. So similarly, how do we query a haplotype from this graph? So let's say this is the haplotype we want to query, that is sample 0. Then we traverse upward to node 6 and node 7. And we know this particular node has has singleton variant M367, which means we will need to put M367 to this haplotype, and node 6 and 7 also has some mutations mapped to it, and then we place this mutation to this haplotype. And this is another example. So node 2 traverse to 4, to 7, to 5, to 6. So all these mutations will be placed to this haplotype as the variant it carries. And the rest of it would be the default, the same as reference, if we define the default as reference. If we define the default as major allele, then they're the major variant. If we define the default as the ancestral, then they're the ancestral. So we could have these different ways, but this is how we're going to extract that information. So you can see that there's this symmetric relationship. If we want to extract the mutation, it's simply traversing downward. If we want to extract the haplotype, it's simply traversing upward. So what are the differences from an ARG? So ARG encodes this genealogical events, and these are the biological events: mutation, coalescence, recombination. And you can think of them as objects. So ARG really encodes these objects. And GRG encodes this hierarchical relationship in the data generating process. And so in that, we only really have nodes as the object. And these nodes could contain mutations. And there are this relationship among nodes that form this graph. So each node bears 3 different meanings. First, they can represent mutation on this node. And secondly, they can represent the haplotypes defined by the mutation on this node and all the mutations on the ancestral node. It can also define a subset of samples as the samples below this node, covered by this node. So it bears these 3 meanings at the same time. And what's interesting is really about this recombination. Recombination is encoded as an event, but then here in the GRG, it's encoded as how haplotype is formed through recombination, and that is through these downward edges pointing to the same node. So you can see that we could convert an ARG into a GRG. So to do that, we simulated some ARGs, and we can have a size comparison based on simulation as the number of samples grow. This is how file size increase. If we have VCF file, it's this line. If we have Plink bad file encoding, it's this one. And this is the tree sequence encoding in this light green color, and this is our GRG encoding. So it has this sublinear growth with number of samples, and is much smaller than any of these intuitive tabular format, and is still in its optimal smaller than tree sequence. But that's really not a big difference, but also not the reason we're inventing this new format. What we're more interested in is whether we could infer a GRG from phased whole genome, and could we do that at a biobank scale? So to do that, we developed a method to do this inference, and it roughly has like 5 steps. First, we would split the genomes into segments, and then we would try to build a graph shape within each segment. This is like trying to find the consensus relationship among these haplotypes and make it a graph based on how similar these haplotypes are related to each other. And then we would map mutations to this graph, and we would merge different graphs from different sections of the genome, and then eventually we would simplify it, number the nodes based on their topological order, and then serialize it in CSR format. So there are 2 time-consuming steps in this algorithm. One is how do we build a shape, because we need to organize the haplotypes based on their relatedness with each other. So to do this, we start with this regular sequence space, and then we use a sparse representation of the genetic variation for these samples, and then we further use Bloom filter to project it into the Bloom filter space. You can think of it as a dimensionality reduction technique or random projection into the integer space, which is really to maintain the relative relationship in this high dimensional space among these among this haplotype at the SNP space, but then it's much lower dimensional and is faster to compute the similarities. And the second thing we use to facilitate this step is something called a BK tree data structure, which is really to facilitate the search of the nearest neighbor from one haplotype to another in the Hamming distance space. So we use this BK tree data structure to find this nearest neighbor, and then we group them into this regular tree space to build the shape of this initial graph. So you can see that this initial graph is built from the reduced space, so it's built much faster, and it doesn't quite represent the data yet. So the next time-consuming step is how we map mutations to this graph. So we have an initial shape of the topology, and we want these nodes to have mutations mapped to it. So if in this example, the first example, if this mutation has descendant 2 and 3, there's already a node that could represent it, then this mutation will be mapped to this node directly. And in this second example, there's these mutations with 0 and 3 sample, and there's no node that currently could contain this mutation, so we will need to create a new node to represent this mutation. And in this third example, this mutation has 1, 2, 3 as the sample carrying it, and we already have a node 5 that is containing this subset of node 2 and 3, so we would reuse this node and then create a new node on top of node 5 and node 1, and then this sort of maximize the reusable hierarchical structure and keep the graph smaller. And then after that, we would do this merge, renumber, simplify, and serialize step as mentioned. So how does it work? So we apply it to the UK Biobank data with the phased 200,000 whole genome sequences, and this is a comparison. On the x-axis here is the chromosomes of different lengths, and on the y-axis is the file size. And so raw VCF, of course, is really large, but even with the gzipped, that is the compressed VCF, we're about 14 times smaller than the gzipped, and so that is pretty great. And this second figure is showing the computational time per node, and we used a cloud node with 70 threads. And so for the longest chromosome, it takes about 14 hours per node. So this can be done quite readily. And the cost for the longest chromosome is 7 pounds. And in total, it took about 80 pounds to do this entire graph construction. So I would say that's very cheap to do. And so you might be aware that there are these more recent techniques that do this highly optimized genetic data compression, so which is a combination of the sparse representation of the variants at low frequency and the PBWT representation of the high frequency alleles, and together with the Z standard compression. So there's these two neural methods, SAVI and XSI, that we compare to. That's not shown in our preprint because we just recently did that. And so comparing with this highly optimized techniques that they do not represent genetic variation in the same way and is further compressed, this yellow line is our uncompressed GRG. That is only a little bit larger than this compressed format. And if we also apply Z standard compression, we can reduce our GRG to the same size as this PBWT and sparse representation-based approach. And so this is a simple test with the UK Biobank chromosome 22. So we can also see the compressed GRG file size is on par with this 2 at about less than 1.5 gigabytes. And if it's uncompressed, it's only 2.4. So we don't think at this level we need to compress, because the benefit of having a graph structure is that we don't have to involve a decompression library when we load in the data. And at this scale, we don't think it's that necessary. So we're not planning to support the standard compression further, because all we care about is to get it down from hundreds of terabytes to a few hundreds of gigabytes. And we also compared with arg inference approach based on the current most scalable arg inference TS-infer. This is done with the 1000 genome whole genome data because it cannot be done with biobank. So the inferred tree sequence is this light blue, and the inferred GRG is the lowest one in terms of file size, and in terms of the computational time, this is the inferred GRG. So at 1000 genome projects, like 2,500 genomes level, we're about 1,000 times faster than TS-infer. And this light blue line is what if we first infer the tree sequence and then convert it to GRG, and that really takes no time because it's just converting one graph into another. And so I want to also illustrate like how to do computation in this new graph structure. So regularly, if we have a tabular data structure to compute allele frequency, we will need to go through each row and do the counting, and then summarize the frequency. So it would be like sequential, row by row. And on this graph, each sample node already has a frequency affiliated with it because that's just 1/n. And then each ancestral node will summarize this descendant node's frequency, so it's simply adding it up. So we would do one round of bottom-up topological traversal to get the value for all the nodes, and then that gives us the allele frequency of all the mutations. So again, this is in the UK Biobank. We compare with if we use GZ VCF to compute allele frequency with Plink, or if we convert this format into Plink and also use Plink to compute allele frequency. And this is if we do it with graph traversal in GRG. So you can see that like compared to this highly optimized binary Plink format, and allele frequency computed by Plink is also based on bitwise operation, so it's really fast. We're still an order of magnitude faster, and which means we're much cheaper and as well. So also recently, just very recently, like last week and yesterday, benchmarked with XSI and tree sequence. So we have very few data points here, but XSI takes about 50 times longer to compute allele frequency compared to GRG. And tree sequence is about the same time as Plink, roughly speaking, maybe a tiny bit faster. But overall, you can see that this new format we developed could really facilitate computation speed. And why do we think it's faster than XSI? It's really this GRG compressed the data in the same way as it represents the data in the memory. So we do not have to convert this format into another format when we load it in. So it's really just like the same thing being used in being saved in the hard drive and being used in the RAM. So it could be accessed very fast. Whereas this PBWT plus compression tools such as XSI, they're still essentially a tabular format, and then they need to take a different representation of the data in the computation, and that makes things slower, even though in terms of size is about the same. So what is GRG really? So GRG is a multi-tree data structure, and this is not entirely a new concept, and actually it's quite old. And a multi-tree data structure is really directly a cyclic graph where each node has a feature that both below the node to its descendants is a tree, and also above the nodes to its ancestor is also a tree. So you trace downward is a tree, you trace upward is a tree, just like what I showed in GRG before. So why is below a GRG node a tree? That is really coming through a coalescent tree. So we have a whole coalescent tree that's for all the samples in the region in the genome, and we're representing this mutation as a node, and below this node is a subtree from the coalescent tree. that pass down this mutation to the sample genome. So it's very intuitive from population genetics perspective why below a node is a tree. So it actually took us quite a while to realize why above the node is also a tree. So we know above the node is also a tree as we trace back, and that was quite clear at the beginning, but why it makes sense that above the tree is a node is really a recent realization after we preprinted. So this is a pedigree data structure to a haplotype. So if we trace backward generation after generation, this haplotype is formed by a recombination between two haplotypes, and this one is formed by recombination between two haplotypes. So if we think about having a recombination event to form a haplotype as this, and tracing back generation after generation, so we sort of also have this tree structure above a particular haplotype. And this haplotype have a few genetic elements. So if we draw lines that's only tracing the genetic elements passing down to this, passing down material to this haplotype, we would eliminate a lot of other haplotypes that's not genetic ancestor to this haplotype. And that also gave us a simplified tree. And that's a tree structure. So a recombination now is really also reflected in this tree structure, but in a much different way from what we think about in terms of ancestor recombination graph. So this is a recombination in forming a haplotype. So it's not necessarily the same as the recombination that happened in ARC, because only if it forms a haplotype that gets passed on to a sample genome, we will record it. But this, again, is that recombination in forming a haplotype. So that's why above it, it's a tree. So now we demonstrated that, that this multi-tree data structure, this GRG is a multi-tree data structure, and we can see that a lot of this graph traversal technique, such as depth-first search and breadth-first search, and this topological order algorithm could be used for GRG-based computation. So we think there's a lot of potential in using it for future whole genome analysis in population and statistical genetics. So in summary, we define this novel data structure to encode the genetic inheritance as a fully connected graph. And we developed a scalable inference method that works at biobank-scale, whole genome level. And we demonstrated it's a multi-tree data structure, and uncompressed GRG can compress the data just as well as this highly optimized genotype compression methods. So with that, I'd like to we have preprinted, although half of this talk is not covered in the preprint. We're going to update it. And our library and the source code are already released. And so I'd like to thank my team, and I have a very small team that's pretty much like 2/3 of all the people I have. So there was like there were some questions yesterday about new PIs, so I was very nervous. You know, like there's these questions raised by students that new PIs don't know what they're doing, and I think this is sort of really like when I started my group, I really don't know what I was doing, and so I'm really thankful that I have a great team, and so this is the result of what we've been doing, and thank the funding source.